Welcome to I Am Nano.
Putting the I in I Am Nano, I am your host, Irfani.
And I am your other host, Monica.
And today we have a special guest to interview as part of our Nano Life series.
Very excited to introduce the grand prize winner of Prix Media Newcomer 2021 from Switzerland, PhD candidate from the University of Basel, Simone Pengue.
Hi, nice to meet you. Yeah, thanks a lot for having me here. What can I say? I don't actually have a lot of time for hobbies or anything besides the PhD, to be honest. So I just try to have some little time to do science communication as I guess we're gonna talk about. In general, things that I like besides science and science communication, I would say it's a lot tea. I'm a heavy tea drinker.
Tea.
Okay. And I'm generally a fairly sport guy, so I'm into weightlifting, running, doing a bit of nothing special. I don't accomplish any result, but it's just to keep myself fit. And I would say that actually there is one thing that takes for me a lot of time besides doing science or science communication, which is still a bit science related, and this is literature. I do quite some literature research. So I'm not just talking about reading books, right? Doing some research about Italian literature, some Italian authors. And what I try to do is comparing some books or some poems with some aspects of science. And I actually started collaborating with the Italian Studies department of our university, trying to do some research on it. So writing papers, going to conferences, this kind of stuff. Actually, there is let's say Italian Studies professor, not my science professor.
Okay.
The people I collaborate with, they find very exciting is that there are some aspects of some books. I can mention one that maybe you also know, which will be the comedy from Dante.
Of course, the Divine Comedy, right? Yeah.
So this has nothing scientific in it. So I'm not trying to say that there is some, modern science in there, but there are some aspects of the way that he talks about sinners, about the people in the hell, that are actually very, very similar to some aspect of quantum mechanics. And so if you really just compare the way he talks about this, the way that the character himself, Dante, is interacting with these other characters down there, you can really see that there are similarities with quantum mechanics. And out of this, you can do, interpretation of the text itself.
Your work published somewhere that we can find it?
Yeah. So for example, this would be on a peer-reviewed journal, which is called Forum Italicum. But I have to be honest, this is in Italian. So it's Italian literature, so...
Okay. Well, we can always link to it in our description, and then we have some listeners from Italy, so maybe they'll be interested.
That will be very cool.
Yeah, no, that's fascinating, actually. And so are you the physicist that's in charge of deciphering and adding the science?
So you see, doing research in the field of literature is very different from research in science. Especially because usually works are not really collaborations between people. In science, we are used to have 3-4 authors for one paper, as each one doing a different part. In literature, you take care of everything. And you only have one person writing the entire article or whatever you're writing.
Do you not discuss with other?
So yeah, exactly. So I have there is a postdoc in the University of Basel that sometimes I discuss my ideas with. There are some people from other universities I get in touch with and discuss with. There is a professor that somehow is judging my work, saying, okay, this is publishable, this is not publishable, just get rid of these. But yeah, so it's a lot on just on me, but the cool part is that I'm totally free doing this,
right?
Because it's not in any big picture of any professor, so I can talk about the author I want when I want. I have no pressure. Right. I can publish whenever I want, and it's just for fun, really. I'm doing a PhD in, so I'm just doing a bit of history so that I will tell you how this literature stuff started.
Yes, perfect.
So I'm now doing a PhD in biophysics in the Department of Molecular Biology, which is called biocentrum. But before this, I actually started a PhD in atomic physics, right? So something that was already quite different. Now, along the way, actually during my first year, I realized that it was not really the field for me. I hated electronics. I hated staying in the lab. So I just decided, okay, it's better if I just switch topic as soon as possible. Now, what was actually cool at that point is that while I was switching from one lab to the other lab, I had a lot of spare time, like 3-4 months during the semester, teaching semester, where I basically had nothing really to do. And I took the opportunity to follow classes that I never took before. And one of these was a literature class, an Italian literature class about this Italian poet called Francesco Petrarca. Maybe you know, I think in English like Petrarch, something like this. And then I was just enjoying it. And then at the end of the semester, I had some idea of, it just came out spontaneously because some things seemed to me very, very similar to an harmonic potential and emotional harmonic potential. I talked about this with my professor. The professor liked this, said, okay, you should publish it. And then I started from there. I really started from, just going there with no expectation, with no goals.
Wow. I wonder what the difference would be. Have you found differences between Italian works and English literary works? Or are you only focusing on the Italian ones from now?
From now, I'm really just focused on Italian works. So I don't know much about English literature. Actually, I would like to somehow manage to reach a broader audience with this work. And for this reason, I think that my next article, which I think it's gonna be ready like in 1 year or so, about an Italian writer who's called Gadda. This would be written in English, yeah, for sure.
We kind of briefly went over that you're doing your doctorate and kind of switched over. So I was wondering if you could explain to us, with your current doctorate, the one that you're doing right now, what's your research focus? Like, what is your thesis on, and what drives you, and what are you doing every day, 9 to 5, Simone?
Okay, so what I'm supposed to do every day, we do nanobiology. So we are a nanobiology lab. In particular, we focus on the nucleocytoplasmic transport. So that's the main, biological question. So how does stuff get in and out of the nucleus of a cell? So the group in general tries to tackle this question from many different perspectives, of biology, of pure cell studies and so on. What I do personally, I try to answer or just put my little piece of answer through the perspective of a physicist, because by training I am a pure physicist. I'm atomic physicist actually by training. So what I'm trying to do is doing a mimic system of the nuclear pore itself, where we just replicated the inner structure of, it's called the central channel, and we replicate it in vitro completely in a very synthetic way, let's say on a plane. Instead of having a pore, we replicate it on a plane. And then what we do is that we label a very few particles, a very few molecules, and then we study at the single molecule level the dynamics of these molecules in different biological conditions, or in different conditions that could be biologically relevant. So you change molecule, you change concentration, you change, maybe you change combination of molecules, and you see how they move, basically. And this would somehow try to answer to some question about how they move inside the pore. So, because, usually when we talk about biophysics, the main current of research is single molecule studies, or one-to-one molecule interaction, usually using a technique which is called nuclear magnetic resonance, NMR. You just take two molecules and you see how they touch each other. This is one end. The other end is cell biology, where you have an entire cell with a huge mass inside, and you see you try to maybe knock down something, you try to enhance the production of something else, you try to change the DNA of your cell and see how this reacts, to try to understand the behavior of each component. What I do is in the middle, and we're trying to bridge this gap. So now, as I said, it's just on a plane having these particles that move, and then of course we'll try to mimic more and more closely the actual shape of a cell, trying to make something which looks a bit more like a nucleus, and yeah, but this is like for the future. Mm-hmm.
Wow.
I also have to say that I hope that in less than 1 year I'll be out. So this is something I really, I really would like to add. So all this, I'm saying from the perspective of a last year PhD student. So finger crossed.
Fingers crossed. Yes, exactly.
Yeah.
We all hope for you. And sorry. So, and why is this research important? Can you touch on that?
Well, there's always a question that is not trying to answer, because it depends a lot on how much you zoom out, right? In general, understanding the nucleocytoplasmic transport is crucial for many diseases. Whenever there are so many health-related issues that are connected to something going wrong in this transport. I would say that the first example that comes to my mind is for sure cancer. So when you look at a cancer cell, you will see that all the actors of this transport are completely messed up. Some of them are too many, some of them are too little. They're not where they, let's say, should be. They're in other parts. So something is going wrong in this transport. But understanding how this transport works is crucial. Mm-hmm. Because you could say, okay, maybe this fixing the transport itself could help curing the disease, could help curing cancer in this case. And now we can zoom in a bit and saying why my work in particular is relevant. But I think, like, if you agree, I would skip this, because this would take a lot of time. Okay. Because it's something as usual, when you zoom into one particular project, I should explain you different models of the nucleus, the different types of transport, and I think this would bore our audience.
Well, they can always read your work online. We'll link that in the description if they're interested.
Yeah.
Yeah, I will link some papers just in case someone wants to catch up on this.
We'll include that. We'll include them.
Go down the rabbit hole of your research.
So going on from here, I think we kind of like touched a bit on it, how you mentioned that the first PhD you did was not pertaining to what you like. It's, you don't like electrical stuff, so forget about that. So that's one of the challenges as PhD student. You have to really be passionate about what you like. You have to just love what you do. And I'm sure that's not the only challenges that you faced as a grad student. Can you elaborate on how you overcome and face the challenges that you had throughout your career?
Well, this one that you mentioned is for sure the largest, I have to say. So, because I was really, what I was doing in this atomic physics lab was Einstein condensates. In particular, we're exploring the Einstein-Podolsky-Rosen paradox in the Bose-Einstein condensates. Super cool topic. This so exciting. It's really just like the limit of quantum mechanics. Okay. And so I was so excited when I joined, but then staying in that lab every day for me was a bit of a torture, I would say. I realized that I was somehow, when you sort of look for excuses not to go in there, everything was a good excuse, and then it actually takes a lot of effort to say, okay, this is really not for me, because that's what I really wanted to do at that point.
Yes.
And realizing that it was maybe a mistake joining that lab was not easy, and for this I have to really thank my professor back then, who's also, by the way, very active in nanoscience. And my professor, Philip Treutrein, at the University of Basel, one day we sat down and he told me, listen, are you really sure that this is a fit for you? Because I don't see that it doesn't come spontaneous to you. It doesn't come natural. He told me, you could do whatever you want, but consider that it's much better maybe changing the topic from the first year rather than struggling for 4 years. Here, PhD is 4 years, and then maybe failing your defense at the end. And this was a very good piece of advice. And also he told me this sentence that I think I will never forget, and this would help me through the rest of my career. He said, you have to do what it comes from natural for you. Because usually people tend to say you have to do what you like, but then you meet people like me who basically like everything. What do you do, right? But I didn't have this natural, natural inspiration to go in the lab and deal with electronics. And then I started doing what was more natural for me, which was this bio stuff. And I should mention, even before joining Atomic Physics Lab, I was unsure if I should have gone for atomic physics or biophysics. There was already 2 alternatives.
Okay.
And I started atomic physics, not for me. Okay, so just refocus on biophysics. And this is something that I'm carrying along also now that I'm somehow starting something in science communication, science journalism. This comes very spontaneous for me. So even though I know that I would like also doing, for example, a postdoc or doing some other type of jobs, they don't come as naturally as science communication for me. Now, to answer your question, what kind of challenges I faced? Well, it takes a lot of passion, but also takes a lot of patience. You really need to, doing a PhD, someone once told me, doing a PhD is pushing your frustration level higher and higher. And this is really true. The experiment doesn't work. Your PI doesn't, just wants result. You are not able to provide result. One of the main challenges that I have personally in my lab is that what I do, since I'm in a molecular biology department, but I'm a physicist, no one really knows what I'm doing. So there are not many people around me that have the technical skills to help me out when I have a technical problem. So I had to figure out many things by myself, which is a good and bad, of course, right? You learn very, very well, but also this takes a lot of time. Yes. And maybe use a lot of time, just chasing artifacts or whatever. It's not you think it's a problem, maybe it's not a problem. And this was, I would say, the main, another main challenge that I have now.
Thank you so much for sharing that advice. Very, very raw, very emotional, and echoes very much with me. And now, so I'm wondering, because it's an over half an hour show, it's freely available online on YouTube. What inspired you to create this documentary for forgotten data, and why that subject? And so I know we're big on the science communication. I know you're very passionate about that, very passionate about science communication. And that's how we found you, is through your documentary, the Forgotten Data documentary that you won your award for. And I noticed also there's some articles about you on how you made a dry subject presented in a super exciting way. Yeah, I'll let you kind of allude and talk about it more.
It's almost silly. It went out like this. I was doing my research on my project, my single molecule experiment that I talked about, right? And I had my primary data, my primary results about the dynamic of the molecules. Okay, all very good and beautiful. But then, while I was acquiring this, the instrument was at the same time acquiring many other information about my data, about my molecules. And then I noticed that in these other informations, there seemed to be a trend. Like when I was changing the biological conditions, these other things, in particular, it was the lifetime of the fluorophore, the lifetime of the synthetic dye, was changing as I was changing my conditions. So it's like, okay, what if I relate it to, what if I find a correlation, right? So I took all this data, I put it into a plot, I showed this to my professor, super excited. I just said, yeah, and so what? So you're showing me this correlation, but are you trying to answer to any question? As no, I just think it's cool. And he said, yeah, you are missing a question. What do you do with this? I see that it's cool, but if you don't know how to interpret, if you are not trying to answer to any question, this data is completely useless, so you cannot do anything with this. In a sense, I mean, you put it down very nicely, I have to say.
Yeah,
but
yeah.
the thing is that he was right. I had an answer, but I didn't have a question. And then I started thinking, oh, but is there someone out there that could do something with this? Mm-hmm. I mean, I put a lot of effort in it. I put a lot of time, and I put a lot of public money into acquiring this data, right? Mm-hmm. I think that if there is another lab that could use my data to do some valuable scientific work, that would be just very cool. Mm-hmm. And then I started thinking about about this idea of sharing data, about this idea of recycling data, and also about this idea of storing data. Because yes, we have our data, whatever it is, we put it on a hard drive, we put it on some server, but then what does it happen to it after 10, 20, 50 years after it is published? Mm-hmm. And at this point, I really just started collecting information. I saw that there was something going on in terms of opening the data, so open data, or there are some programs in some research labs, especially private research labs from pharmaceutical companies, recycling this data. So I thought, okay, I should maybe put all this piece of information together. I mean, I think it's a very important topic today. It's a very important topic because producing data costs more and more money. Mm-hmm. It's costing time. Yeah. We have the technology to go through this data in large data sets, which is artificial intelligence, for example. Mm-hmm. Mm-hmm. So we would be able to do it, but of course it's not easy. Of course, it takes a lot of efforts. It takes a lot of coordination. And at the same time, we cannot just store everything all the time forever. That's also very costly.
Right.
Yeah. Mm.
Yeah. Perfect. It's true. Yeah. It had a lot of questions that I received once I was like looking at your, I never thought of this. Oh, that's brilliant. Oh, interesting how Roche does it. You also touched on that during your, like big pharma.
Yeah, because you see, actually, one strong characteristic of companies, in particular of Roche, is that there is a central coordination. So in the film, what we show, so we maybe it's useful for those who did not watch it yet, that I could maybe just go through the main steps, right?
Yes, perfect. Go
so as I said, we are just trying to answer this question, not really answer to any question, or to be honest, we don't answer to any question. The main goal was just to open the debate, see that it's not an easy topic, but it's an important topic. Mm-hmm. So at the beginning, we talked with Michael Podvinets. He's the data manager of my institute from the Biozentrum. He was actually, to be honest, one person that a few years ago put this seed into my head, because he was the person who trained us on how to store our data, how to manage our data. And he started talking about this idea of storing for a very long time, to giving ID to your data so that maybe other people could access, talking about public repositories. And then that's really the moment where I really started thinking. This was at the beginning of my PhD, so 4 years ago.
Okay.
And he's the first person that we talked to, and I think he's a great speaker in general. By the way, there is some German speaker out there. Check out his science and cuisine books. Mm-hmm. I think they're amazing because he's an amazing science communicator. He's also a very enthusiastic cuisine lover, high cuisine. So he tries to connect this. So anyway, getting back to the film. Okay. So this is the first person we talked to. So what does happen to the data? And then the second is this Maria Grazia Giuffreda. She is the vice director of sorry, I just know the Italian name is called Centro Svizzero di Calcolo Scientifico. This will be something like Swiss Center for Scientific Calculus, I think. So they have these huge supercomputers, and she's the vice director of it. And she explained where the data are actually stored, what are the different possibilities of storing them from long term, like, on the facility perspective. The third speaker is Professor Michaela Zavolan. She's a bioinformatician, so she explains the way that she extracts information out of large data sets, how she uses them, what can a person usually do, what is the field doing in this direction. Then there is the contribution that you mentioned from Roche. Roche is one of the largest pharmaceutical company in the world. I think it should be like top 3 or something like this by very huge. Actually, the skyscraper that you see, it's in the movie, that's the tallest skyscraper in Switzerland.
Oh, yeah. Oh, and it belongs to the
belongs to Roche.
Wow.
Yeah.
Yes.
Okay.
It's just in Basel. What I like there, and this to answer to your question, is that they recycle data quite a lot. They recycle data much more than the academia, for 2 reasons. First, because they know that the resources are very limited. I mean, they have large resources to do research, of course, but they value them very much, and they hate repeating the experiment twice. So they want to gain the most out of one single trial, one single data set. This is the first thing. And the second, they have a central coordination. And this is what is really missing in academia. What is really missing is someone that says, oh, you are looking for this type of information, this type of data. Just ask this person, they have them. They did this experiment very, very well, but they didn't publish it. Or they published the paper, but did not publish all the data for some reason. So ask them, and you would save time, right? Time, resources, energy. You can make a great collaboration out of it.
Yes.
And if you think about it, this is the same thing that happens in an individual lab. So in one research lab, you have one PI with this central coordination. And whenever you want to do an experiment, maybe it also happened to you, to me it happened many, many times. I want to do this experiment, and he told me, oh, but ask this other person, he or she did this. It was many years ago, I don't remember the outcome, but we have it stored somewhere. And then I save a lot of time and energy.
Yes. Yeah.
And we put it together. And then the last person we talked to is a bioethicist, David Shaw. Also from the University of Basel, but he was in Glasgow at the time of the interview. He's clearly Scottish, right? He's clearly Scottish. And he explains the problems of sharing the data, which is not always easy. It's not easy on a facility perspective, on a facility point of view. It's a lot of data. It's not easy on an ethical point of view. It's not easy on, for example, in case you have sensitive data of patients, because we have to a bit zoom out sometimes, right? To remember it's not just, for me, just biology or just nanoscience, but you can also have other fields. What could be hard in terms of intellectual properties. So if you use my data to do a paper, am I mentioned as an author? Am I just acknowledged? Am I even mentioned? It depends. It depends, right? It's not easy.
Yeah.
And then he also, and this is one thing that I like the most, he also brings up the point that we just cannot store all the data all the time. This also takes energies. It takes electricity. Yeah. So I don't remember if this answered your question.
Yes, it was perfect. And I'll let Irfani go and ask hers. Yeah. I know she has quite a bit for you.
So you mentioned that in academia, we don't have the same type of organization of data like Roche, because they have a repository with everything that they've done, and then they reanalyze the data. And I think you mentioned that in the movie, maybe you're extracting data from different contexts and then see if you can extract a different new knowledge about that So it's not necessarily answering the question that you had in your first experiment, but if you see a pattern from different experiments, then you can, gain new knowledge from that without having to do a new experiment, right? So
yeah, that's very correct. Maybe you are trying one molecule, and you're testing one new drug onto some voluntary patients or some developing a new treatment, and you see that on some people it works, on some others it doesn't work.
Right.
And okay, maybe you then don't release commercially this drug, but you still have a lot of information, and you see that this drug did not work. But hey, also this other drug that you tested 4 years ago did not work on the same type of patients.
Mm-hmm.
So now if you cross-correlate the two, you can see that there's a pattern, and the reason why it didn't work on this type of patient was because they have some other conditions behind.
Right. So it looks like they have like a very good organization about their data and storing their data, and that's something that we're really lacking in academia, especially, at least from my experience. Every single lab have their own little group, and we don't really share our data with each other that much unless we're collaborating. And even in my own group, we have multiple projects, right? And then we have completely different people doing completely different things. But the one thing that I'm also noticing is it depends on the supervisor. Some supervisors tell you how to store data. So in your lab notebook, they tell you how to write the data, so then future students can refer back to it easily. But for me personally, I have my own way that works for me, but it may not necessarily translate well for the future students. So when they look at my notebook, they may get confused. But it's like it's not consistent, and it's not a system that's very easy to translate for the ease of sharing information with other people. So I'm just wondering, like, how do you store your data in your lab, and do you make sure that it's easily translatable for the future students, or like how does it work for you for your own experiments and data?
So to answer this question, actually, we have to talk about a key concept, which is metadata, right? Mm-hmm. Data about data.
Right.
It's not just a matter of having the outcomes of the experiment, but as you just mentioned, it's also about all the information about the experiment itself. In which condition did I do this? Which instrument did I use? Which was, I don't know, the settings of the instrument. As you a priori, you don't really know what is relevant, so you have to include as many information as possible. But also, if you try to give this data to someone else, many things you gave for granted, but are not granted for this other person.
Right.
Not at all.
Mm-hmm.
And without all this piece of information, the data is just useless. Is completely useless. You cannot do anything with it. And I think that Michael Potvinets in the movie explained this very, very well. Metadata are really the key, and they're also the largest challenge in terms of when it comes to sharing data. What we do in our lab is something that I don't know how common it is. We have a shared lab book. Not really shared. I'm going to say it's just online. Everyone can access each other's lab book, and on this lab book, which we call the wiki, we can just write as many things as possible, and is organized in a nested way. So you have your project, you have the current focus, what you're trying to answer to right now, why you did this experiment, and you try to put as many information as possible. So this is what we do in our lab. Then, of course, we store the data on some servers, and these data are actually not accessible for external users. These are just for our lab. And one key point, and this is always a tricky point for every scientist, is very important to name the files properly. Just the file name is data itself. Don't just write a code, just write data or test or a just write many things, write as much as you can. And this is also a crucial thing. Maybe from this I can also answer. I can also expand this topic a bit, saying what we do with the data in our lab, in general in Switzerland, because as I mentioned before, the idea of recycling data, of sharing data, is becoming more and more important. In Switzerland, the funding agency, the SNF, they demand that the data that you publish is then accessible for external users, because the idea is that it is publicly funded, so everyone should access them.
Right.
It's also sometimes demanded by the papers themselves. On some journals, when you publish your paper, they ask you to share the data on some public repository. Here in Europe, there is one which is handled by CERN, which is called, I think, Zanado or Zenedo, something like this, which is very popular. And you just drop your data there. It has a permanent identifier, and then you can directly share this data. Now, this is, as I said, very recent. In a sense, just 2-3 years that is small spreading around. Is relatively, I mean, it's just doesn't take much time, because you really just upload your data there, and then you're supposed to explain the data in the paper. So usually the two things are connected. But what we are missing, what we are lacking of, is recycling this data, so really reusing this data. The main idea behind posting the data somewhere, the original data somewhere, after your publication, is to prevent accusation of fraud. So the idea is that, okay, you can take my data, reanalyze it with your own software, and see that I'm not lying, see that I'm saying something reasonable. And what we don't have yet is many people using other people's data to do new science. Or better, I'm not saying that this never happens. I'm not saying that this is just doesn't catch us up. I'm just saying that it's not super common. It should be more common. And the process right now, it's also quite complicated. Me, myself, I tried just a few weeks ago. I had this new algorithm to analyze my data from my instrument. But nothing super fancy. So I'm not doing a publication out of this algorithm. It's just a few lines of code. I wanted to test it. I wanted to see if I can claim what I'm trying to claim using this algorithm, and just using my data or just producing data ad hoc maybe will not be the best solution, because I might introduce some bias. I don't know. I want to make sure, right? So what I tried to do, I tried to go on some repositories and download some data of people using the same technique in very simple conditions, and try to see if the algorithm was work. Actually, I failed. I could not find a single data set that was useful for me. Honestly, my demands were very, very easy. I mean, I didn't ask for much. I just wanted something simple. Now, what I tried to do was both going into these repositories and directly search with some keywords, or going to papers and see if they had some indication. You can find the data here. But the problem, as I said, is that this is just a new trend, and not everywhere, not in every journal. So the most of the papers, the data was just not available. In the repository, on the other hand, the problem was that the keywords were just too general. And I was always ending up with something that had nothing to do with the simple things I wanted to do. And to be honest, what I needed was something that, a bachelor student does for a 1-week project. Just take 2 proteins labeled with the same dye, put them together, put them in front of this type of microscope, which is called fluorescence spectroscopy, and see if you can distinguish the two. That's everything I wanted to do. Super standard.
Yeah.
But I didn't find anything.
Yeah, and it was probably there, and it was probably done multiple times, but just mislabeled so improperly that it's hard for someone else to find.
Exactly.
Interesting.
And this is the largest problem right now. Maybe something is out there, but then you also have to find it. So what you need are domain-specific repositories, and these are super helpful. These are super helpful because you can find information directly. They are very much standardized. You know where is what is where all the time. You can just download it, and it's ready to use and ready to be digested by your machine. And this, for example, I mean, this one thing that I think is key here is distinguish between published data and not published data in terms of paper, because also sometimes data that is not good for a publication can still be very useful.
Yes.
Right. Yeah.
And let me just bring one example. Just one example. This example would be this recently released algorithm, which is called AlphaFold. It's a do you know it? Have you ever heard of it?
AlphaFold, like for proteins?
Exactly, for proteins.
Yes.
So everyone in the protein field knows it because it was a big revolution that just came out a few months ago. So it was basically released by Google, our company owned by Google. And what it does is that it uses artificial intelligence to predict the structure of a protein.
Wait, okay. Wait, is this the time when basically you figure, I think a few months ago, not a few months, maybe a year ago, a bunch of computer scientists figured out how proteins fold. I think they did it. Yeah, it was in Google. It was in London, I think, in UK. It was a huge deal that it worked.
Yeah.
It worked. Are we talking about the same thing?
I think so. Because it's a long-term challenge.
It was never solved, and it was computer scientists instead of biologists to figure it out.
Yeah, exactly. So the thing is the following. So a protein is a sequence of amino acid, right? And the function of a protein heavily depends on how it folds, the structure, three-dimensional structure that it takes, the shape of this protein. What we usually do is that, I mean, it's a huge field of research determining the structure of a protein. You have cryo-EM, for cryo-electron microscopy, for example. It's a very important technique. You have, for example, X-ray crystallography. These are other important technique. So it's a very big and very important field of research. But people wanted to know if it was like mathematically possible to derive the structure a priori. So you just give me the sequence, I give you the structure of the protein. What the scientists, as you said, computer scientists did is that they used artificial intelligence to be able to predict this structure of the protein. Now, what they did is, I think, is a key example in terms of sharing data, because they went to these databases, online repositories. They downloaded a lot of data of structures that were solved and structures that were, also published. So, okay, good data, good type of things. And this was used to feed the machine, because you need a huge data set to feed the machine. Took this, but also they took the data of unsolved structures. They took some data that was not published. I mean, it was released, it was open accessible, but was not a publication because people failed. They said, okay, this is what we did. This is the structure of the this is the sequence of the proteins, but we don't have the structure because it's too hard to get. And what they did, they tried to predict it as well. They gave it this to the machine in order to train the machine. And now the algorithm that came out of this is so helpful. It's so helpful, it's so strong, it's so good. It is an incredible story of success.
Yeah,
amazing. It's very
it's huge.
Oh.
That was a huge discovery to be made, because it's because also with computer scientists, I guess they think about things in a little bit differently, so they're able to kind of reorganize all the data that has already been available, and then just being able to predict the protein based on its sequence. It's huge.
It respects the idea of fair data,
right,
which we talk about in the film,
right,
and it's a rather complex subject.
Yes, yeah, and then there were some bioethical also concerns with that as well, which is interesting.
Exactly.
Yeah. I mean, I don't know about you guys, but sometimes when I'm doing an experiment and then it didn't work, sometimes I wish someone just told me that that wouldn't work, so I don't have to waste my time to do the experiment. So I wish it's more the norm, or more of the norm, for people to just also show the things that don't work.
Exactly.
Yeah.
But that's not gonna end up in natural science, you know.
Exactly.
Yes.
That's not the goal for most people. On the topic of publishing data, are publishers like showing the metadata, not the metadata, the raw data with the metadata? Is it common to show all of these details, or is it common for publishers to just make it freely available? Like how are they feeling about this whole data sharing? Because they're pretty proprietary, because have a paywall behind journals already. I'm just wondering, what do you think publishers think about this data sharing?
So now that you're bringing this up, I think that I should have maybe interviewed also publisher, because their perspective is also very important, right?
Very different too,
yeah. So this is something that is definitely missing in the film.
You don't have to tell that.
But so we somehow talk about this with David Shaw, and he correctly says that it is heavily field dependent. There's not such a thing as a one size fits all when it comes to data sharing. Now, I just bring some examples here. So yeah, the first thing that I should say, I want to state very clearly that it is heavily depends on the journal. Some journals force you to publish your data, some your raw data. Some journals, they just don't care. So this is the first thing. Second thing is that heavily depends on the field. Now, let's start from the extremes, right? On one hand, we have high energy physics. In high energy physics, they share everything. There are people doing their entire PhD, their entire research, the entire lab based on reanalyzing data. Because this data that is, for example, produced at CERN, and then in the US they have the Fermi Lab, which I guess is organized similarly. This data costs so many money and so important, and there is just one instrument in the world that you want as many people as possible to extract information out of that, because you just have one source. And so they are used to these people, they invented the internet. So they of course they are very open-minded when it comes to data sharing. On the other hand, you have something that is not science but still research, which is literature research. Not necessarily Italian. I think this goes for every field in the humanities, where people not only they don't share anything, but the journals are not even accessible online. The field is not even indexed. If you now try to Google my work about Petrarch, you will not find it. Even I could not find it.
Oh right? My goodness.
You have to know that is in that book. It's because the field is not indexed, you don't find anything on Google Scholar. And you see, on one hand you have everything is immediately accessible, on the other hand you have nothing is ever accessible, forget about it. and biology is somewhere in between. Why biology is in between? Because on one hand people are the computational perspective is coming in with a lot of force, and we would like a large data set to train our computers to be analyzed and reanalyzed, and is gaining more and more importance. But on the other hand, the data is not so standard. This depends on too many things to be digested by a machine. And also there are a lot of money involved. So biology, and especially pharmaceutical sciences, they generate a lot of money, so you don't always want to share everything. And then if you move towards medicine, then you also start having the problem of privacy, for example. So the data itself does not want to be shared because the data is a person. And you see, it really depends on the subject.
Yeah. It's very interesting actually that you mentioned the publisher, because also in Canada we have, if you're funded by the national agency, you're required to publicly release your publication or whatever your article, but your journal's through a paywall and then you sign the rights away to your research, right? So who owns your research in the end, right?
Yeah. Well, I mean, on the topic of just like sharing data and bioethics, especially for the medical community, just sharing data in general, especially in the age of social media and the internet, and especially in the open sharing, unprecedented time during COVID, when scientists were just like, here's all my data, analyze it, and then let's work on this problem together. I mean, there are some ill-intentioned people who might cherry pick certain quotes from your article and try to fit it into your narrative, even though it was like a completely different discussion. So that would be like another concern, I guess, if you are making your data public. If you don't have enough knowledge to analyze the data properly, they might take it out of context and twist it around. How do you think we can try to prevent this kind of issue?
It's a really tough question. And I mean, it's, I have to say, to be honest, I don't think there is a clear answer. So this is the famous problem of the dual use. You do something with the best intention of the world to help people or whatever, and then someone takes the very same knowledge to, do something bad. Or, this, I can say, is so unpredictable that there is no concrete way to prevent this from happening. It depends a lot on the scientist himself or herself. This person should know, okay, is this, what can I think about some potentially harmful way of using my data or my paper? Unluckily, I really don't think that there is an easy solution at that point. I think it's one of the main concerns of knowledge in general, not just of data sharing. If you look at the story of technology, well, the most clear example I would say is the atomic physics and nuclear physics. And they did a couple of bombs with it, and now we are in all these problems, right? Yes. Of course, they started with a very different intention.
Right.
But the very same theory that led to the atomic weapons, to the nuclear weapons, they also brought to a lot of good insights in medicine. So they're also saving many lives, the very same papers. What can you do with this?
Yeah.
I'm afraid you can't do much.
You can, yeah. Yes.
Yeah. Well, I think with the rise of, science communicators, I would like to think that we have the best of intention. So we would try to disseminate the proper science, and then if, for example, someone out there is kind of twisting the data, then us as science communicators would be able to like call them out, and then we would, as a collective community, be able to manage that to some extent. That hopefully, in my opinion, one way to tackle the problem, but it's a complex issue, like you mentioned. Yeah. And I guess the last thing that I wanted to ask you, that we've also touched already with the concerns of storing data, is just the sheer amount of data to store and for how long. And we did do an episode a while back about storing data using DNA, which is like in a small tiny bottle, for long-term storage, which is a possibility. I'm just wondering, do you know anyone who's tried alternative methods? I mean, the DNA one is still experimental. It's not in use effectively right now. But I'm just wondering if you know anyone who has attempted to use alternative methods of storing data that's more environmentally friendly.
To be honest, I'm not really aware of technological methods to store data that are more environmental friendly in general. I think that what we should really work on is what we store, for how long we store, and how we store, not in terms of the technology, but in terms of, as I said, making this available. So if right now I take all the data that I took from my PhD and go to one of these repository, very journalistic repository, and upload everything, these are hundreds of gigabytes of data, and there's nothing stopping me to do that. Nothing. The owner of the website will just allow me to do that because that's their job in a sense. But this will be extremely useless. It will be a huge waste of resources, right? So rather than, I mean, yes, it's very important to develop technologies to store data more efficiently, but also will be very important to decide what to store, put in the right place so that is accessible from people in the field, and give metadata around it, organize in such a way that you maximize information, minimizing the space. Even just having some scheme, for example, some table to fill with the data, organized in some systematic way that is the same for everyone within your field. That is much better than a long text. Saves you time, saves space, and saves energy also to the reader and the person that has to pick up that data. I don't really know more efficient way of storing because, as I said at the beginning, I'm not a huge fan of electronics.
Well, you mentioned the data that you collected throughout your PhD is like hundreds of gigabytes, right? But then the one that inspired you to, make this documentary is, that interesting other data that you got from your machine, right? What if that data, say, if you didn't notice that pattern, and then you regard it as one that is not useful, then another scientist who looks at it is like, oh, that's an interesting observation. So how it's kind of hard to like make. So I guess for some data it's easy to decide, oh, that's not useful at all. Or some other data, it may be something that other scientists might be able to see as useful, but we may not be able to see as useful. So it's kind of like hard to sometimes to define it.
Of course, there is some. It's I wouldn't say it's a bet, but I would say that there is always some risk involved. Right. What if I upload it and then some experts looks at it and say, "Oh, this is bullshit." No, in general, you don't know a priori, but for sure, if you just put it out there as randomly, what are the chances that someone is gonna spend time to dig through your data, understand what you did, understand the conditions that you did it, and put in a broader context? The chances are also extremely low. Right? So, yeah, I'm sure we are missing many things out there, but I'm also sure that the most of the data now uploaded in the repository is completely useless.
Since you have access to this repository, right? Would you be able to go to them and say, oh, I'm doing this research, and then if they've been labeled properly, the data from like previous work, all these research data, would you be able to like ask and extract these data sets prior to you doing any experiments yourself?
So as they're currently organized, I don't think that the managers of the platform themselves, the people that are running the platform, will be able to do this, because usually these people don't have knowledge in your biological or nano research. They have more IT type of knowledge. What I think could be, there are two things that in my opinion would be crucial. First is keep talking about this. We have to start a debate. We have to start coming up with ideas, with criticism, with needs. I don't know what you need because we never discussed about it.
Right.
I don't know what you would like to see in a website like this because we never discussed about this. If we just start having the discussion, and it is happening now, but it's the real time to push into this direction. I mean, that's the first thing to do, for sure. There are many associations all over the world that are doing a great job for this. I know a Swiss one, which is called opendata.ch. They are let's say, opening the debate here in Switzerland to sharing data in a much broader perspective than what I am doing. And this would be the first thing. And the second thing is that, in my opinion, we would really need to think about an easy way to access this thing, like easy in terms of, at a social network level. So the one way that I would personally see, maybe this will not make everyone happy, but I was just going to pitch my idea, like a rough idea of it. It's just doing something organized a bit like Reddit, in a sense, where users themselves can, open subreddits. So open some, the little branch of the tree, getting more and more focused onto something. And this user generated, because only the users know what they need.
Yeah.
Right.
So what I would do now in something like this is I would go into the FCS directory, Fluorescence spectroscopy, which is my technique, go into the other directory, which is protein. Then I would go into this directory and open, if it is not available yet, a lifetime directory and post my data there. And then someone that is looking for lifetime of fluorescent molecules that are associated with proteins that are analyzed with FCS, know where to find them.
That's perfect. Yeah, very smart.
Right? And then having also tags maybe so that you can easily research. Because what I saw when I was looking for data that I could use is that every sort of keywords that was coming to my mind were just bringing too many results that had nothing to do with what I wanted. It's not well indexed, in my opinion, right now, this field.
Nika, do you have any other questions?
So just to close up on your documentary. So to anyone listening, and maybe they're inspired by you, they did about your documentary, your movie. How do you make it? How do you even start? And should someone have a timeline and budget? How do they put it out? Just briefly. You're inspiring the next generation of scientific communicators with this, for sure.
The logistics of making a documentary. So there are three things, or maybe more. Okay. Let's not number them. Okay. There are some numbers of things that are very important. First, having the help of someone with the skills. So someone that is very good at using the camera, very good at editing, very good at sound engineering, lighting. This for me was my brother Lorenzo. I really have to share. Yeah, he's just 20. He's just 20 years old. Yeah, and he's the person that was actually behind the camera. Actually knowing how to use it, editing the software, editing the video. So what I was doing, I was really just directing. So saying, we need this, shoot this like in this way and this way. We have to edit in this order and so on. And he was the person who actually putting these pieces all together. So you need someone that is able to do this. And I was lucky because I said it was my brother. Second, you need, I would say, the overview before starting. You need to know what you want. Before shooting the very first second, we already knew like how to develop the film itself. Because if you just go out there with the camera, shoot, then you go, in front of your editor software, and you don't know what to do, and you realize that the vast majority of what you did is just useless. It doesn't stay well together.
Sorry, if I can ask, you mean like shot by shot, like how you want to look on camera?
Not that specifically necessarily, but for example, I don't know. Okay, I wanna interview this professor, right? I know that. Okay, I will go there with two cameras, and I'm gonna place these two cameras somewhere to alternate between the two cameras. Good, fine. Okay. What do I expect to have before the interview with the professor? Like the shot just before. What is it? Is it another interview? Is it me talking to the camera? Is it just some landscape? Is it someone in the lab pipetting with a voiceover? Is it just some music? You need to have some rough idea of this. What we wanted to do, for example, this idea of following basically me going through this, let's say, journey, going to one place, from one place to the other place. So we said, okay, before every interview, there is a scene of me going into the building to meet this person. And that's what you see. in some way or the other way. You need to do this, and you need to know this beforehand. And this brings us to the third point, which is you need a very good organization. We have, so just in terms of timeline, just to give you a rough idea, January, February had the idea. March, I got to know about the contest. April, we pitched our idea to the contest, we applied to it, and March, April, I started getting in touch with the people. Okay, would you be potentially available for this thing? At this point, I didn't know the timeline of how the movie would have been at the end, but I just knew the perspective that I wanted to put together. Now, we got the confirmation in June, and we started shooting in July. So we spent the entire June and July just planning, because in the moment that you have two people there with the camera, and you have the interview to make, and you have the B-roll to make, and you have other people involved, you have the translation. So we had 4 translators for this, right, because we have many national languages in Switzerland. So we had 4 translators. We had many people collaborating with the science shot and scientists here and there. And all this has to be scheduled. And this takes a lot of time and a lot of energy. So this is the third thing that I would say you need. 4, it's ideal if you have, this is really just an advice. It's ideal if you have a context. So when you get in touch with someone because you want to interview this person, especially if it's a professor, someone whose image is somehow precious, you need to specify why you want to interview this person. Okay, I want to do documentary. Okay, who's producing? What's your goal? In which context? Where is it gonna be broadcasted? Who are you working for, basically? This is very helpful if you have, in my case, a public institution behind you, which would be the Swiss Academy of Sciences that was funding me. And because these people trust this, and this is helping a lot. And besides this, I would say that in terms of budget, if we really want to talk about practical thing, we budgeted, so just talking about in terms of numbers, this institution, the Swiss Academy of Sciences, gave us 3,000 francs, which are a bit more than 3,000 US dollars, like I don't know. Yeah. And this was the starting point, but we knew that the price was at 4,000 francs. So what we did, okay, this was a bit of a bet. We said, okay, we're just gonna budget 7K. Okay. If we lose, we're gonna lose 4K. If we win, we're just gonna break even. Oh my God. And that's what we did. I mean, it was, we had no other option because we were really running short in money. And so so this in terms of, to do everything like this, we spent in total 7,000 francs, excluding any sort of compensation, let's say, for my time. So I didn't get a single cent out of this in terms of, salary, of making money out of it. And also my brother, he did not make money out of this. So, and also the gears, like in terms of the cameras and so on, the most of the things we already had, we just had to buy a few more things. So this, I would say that to do something like this, if you really keep your budget low, you can get away with 20K around it, more or less.
Oh, wow.
And I would say that these are the most important advices, like very pragmatically, right? So I have this broad view before starting.
Right.
You need to know what you want to say and how you want to say. Identify your target very clearly. So who are you talking to? What kind of language are you going to use? What kind of flavor are you going to give to your movie or a podcast or article, whatever? This is crucial.
Okay.
I mean,
I'm glad your bet went on, but your bet succeeded. So congratulations.
The documentary was really well done.
Thank you.
It was
Thank you.
really well done.
Yeah.
I was watching it in the car on the way back from like a trip, and I was just like, wow, I was intensely watching it. Like this is something, like it was captivating. It was something that I know, like, oh, we have a lot of data. Yeah, we have a lot of data. But it's like the way you tell the story was engaging, was very engaging.
Thank you.
Yeah.
This
Actually,
was a challenge. Yeah.
Yes.
Yeah. Is there anything else you'd like to share?
No, I would say that I shared a bit everything. Maybe the only thing that I want to add that is that, doing this was extremely stressful. I don't know, like some people that know me.
It was amazing. I could not tell you were stressed out. It was very smooth.
Some people that know me well, they say, okay, in this shot, you're so tired. It's like, yes, because all of this, it just went on top of my PhD.
Right.
I did not take any time off, except for the days of the interviews.
Yeah, which was like 5 days or something throughout,
yeah, exactly,
right?
Exactly. 5 single days, but I mean, it's like 3 months of work.
Right.
Yes.
And this but I had a lot of help. Yeah, definitely. I mean, we really, we in general, me and my brother, we are very happy with the result, and it's opening many doors, I have to say now, in terms of science communication, in terms of networking, people that we got to know, opportunities that we are, currently having still now to start our career in this direction.
Perfect.
Or at least myself, or also my brother as a filmmaker. And also one thing that I really want to say now is that you cannot do something like this just by yourself, right? You need the help of many people that I am extremely thankful to. They are all credited at the end of the movie. But besides my brother, who, as I said, shared the 50% of the workload, there are other, let's say, 3 people. In general, there are many people, but 3 in particular that helped me a lot during the various processes. 2 are 2 very good friends of mine. One is Florian Derbar, he's a PhD student in my institute, and the other is Nicole Maniero. She just graduated from Cambridge. She does Italian studies, by the way.
Wow,
and then the third is my wife, Laura, and she also helped me a lot, not just supporting like morally me, but also like more practical things. She was really helpful, and so I really have to mention these 3 names.
Yes.
All the support system we have. Yeah.
It takes a village for sure. Yeah.
Yeah. If I can ask one more question from that. I mean, this was, a feat. The documentary was amazing. Do you plan on like any future projects in communicating science that you'd like to share?
Yes, a lot of things.
Yeah.
There is a lot. So right now, that I'm really working on right now, there is not much. There are some science communication articles that I'm writing, but are a bit, for institutions and this kind of stuff. So it's a bit more boring in a sense, right? Less exciting. There is one more literature work that I'm working on right now, which is about this Freitag Gadda. It's going to come out, I think, next year. Hopefully it's going to be in English. I would like to talk about this connection between science and literature.
Yes.
To a larger audience, without being too specific, without being too academic. And I hope that I'll have the opportunity to write about it, hopefully in English this summer. So that's a bit my planning. I'm just trying to pitch the idea to some some magazines online. And then in terms of documentaries, yes, my brother and I are actually planning on many themes. There is one that would be like, for example, there are 2 main projects that we would like to develop. They are all very, very immature right now, so it's just ideas. One would be, exploring this idea of the connection between art and science, but not art based on science as it is usually presented. or not scientifically analyzing art. This is not the goal. The goal would be to really look at the way that scientists work, look at the way the artists work, and say, oh, but you see that you use a different language, two different languages, but at the same you are saying something very, very similar.
Mm-hmm.
And this would be one of the first ideas that that honestly we need decent fundings for this.
Yeah.
Because this is something that we're going to do after my PhD.
Yes.
So, of course, there's going to be like economical support is going to be absolutely needed. And for the planning, we did a rough budgeting in terms of people we need, in terms of equipment we need, in terms of time that we need. We are around 100K. Without, it's not as 100K that I put in my pocket and become rich. Just this is what you need. 3 months of work, multiple people on it. That's the budget you need. Mm-hmm. So I really hope that we're gonna manage to find this money. It will be one, and another super fresh idea that I'm having recently would be something related. I want to explore the idea of the errors in science, not just mistakes, but the idea of when every time that scientists make a claim or publish a result, they know that there is some uncertainty around it, right?
Yes.
And the role of science communicator is to also communicate this uncertainty, this error. But this is really tough. So I would like to, really focus on this and try to explain to the general audience, all this, how can I say, I would say these error bands or this smoke that is around one discovery. Yes, scientists prove that, eating tea is very good for your health, but there are huge error bars around it. Right. So how do you explain the concept of error bars? And this is something that I think we should insist a bit more as science communicators.
Wow.
Yeah, for sure. I guess
I'm looking forward for all of these.
Yeah, more from Pengue Productions, the Pengue Brother Productions. That's what we want. Yeah, that's what is needed. And I guess that reminds me, because when you accepted your award, you had a quote that is very similar to what you just said. You're quoted saying, "After all, communication about science means not only saying good things about it, but also doing so effectively, understandably, and above all, elegantly." End quote. And I think that's fabulous. It's really a great thing to kind of close off your amazing documentary. Thank you for coming on the podcast. I guess the very last question, we very briefly, because we're a nanotech-based podcast, where do you see the future with nanotechnology? It can be in data, in your research.
Just everywhere. Nanotech are just exploding. I really think it's the best moment to do a PhD in nanotech. To be honest, it's just here in Switzerland is growing so much, also on an economical perspective. So, and I would really consider I will really advise you all to look at, what we do in nanoscience here and eventually planning on a postdoc here or whatever, because it's growing so much, especially in the world of biotechnologies. So where do I see it? I don't know where, but then it's going to be very, very bright.
Very, very. That's good. That's always good. I'm sure yours as well. You can do lots of very bright documentaries.
Thanks a lot for all your nice words and for having me here.
Well, thank you for sharing your knowledge. This is amazing.
I'm really honored to be here, to be hosted by the two of you, to be part of your great podcast. And so thanks, really.
You're famous in North America now.
Yeah. We're going to share it with everyone. I think it's a conversation we need to have, and we're going to spread this word around. So we will link in the description to Simone's documentary and all, his literature, other literature, and his PhD project as well in our description below.
Yeah.
And so I think that is all the Nano for today. Take care.
And stay curious.